Today we will be walking through the process of how glucose is not only able to make ATP, but is also used in the production of nucleotides, ribose, and very importantly, DNA replication. We will start with a glucose molecule and follow it through the various pathways. I'll try to give a quick summary of some initial glucose metabolism, but we'll also see how G6P, a product of glucose metabolism, is used in the formation of nucleotides. There are going to be a lot of different names and pathways to follow, so at first look it is pretty confusing, especially with the overall map. But to help organize things in our minds, I color-coded the first 4 pathways that we will encounter. I find it helpful to imagine where these different processes are happening in our cells, so I organized the pathways based on the location that they take place. Yellow will represent the process happening in the cytoplasm. Orange will be when the pathway is in the mitochondrial matrix. The bright red represents within the inner membrane of the mitochondria. And finally, dark red means that the process is happening in or embedded in the inner mitochondrial membrane. Along with color coding, there will also be an overview of the metabolic map that I have drawn between each specific pathway to help us become more oriented of where we are and where we're going to be headed next. With that in mind, we're going to start with the glycolysis, which is highlighted here in yellow, signifying that it happens in the cytoplasm. We're going to look at a more zoomed in picture here so we can see it better. Glycolysis will start with a single molecule of glucose and will pass through 10 catalyzed reactions until it reaches the product of 2 molecules of pyruvate. These 10 reactions are further divided into 2 different stages, which help us to track the amount of ATP consumed and generated. These 2 stages are termed the energy investment and the energy payoff stages. To save time, I will only mention the steps that are actively contributing to the amount of ATP being produced or generated, or consumed or generated. Also, I will be talking about the electron carriers that will be produced here. In step 1, where glucose is turned into G6P, an ATP molecule is used. In step 3, where F6P is turned into F16BP, an ATP molecule is also consumed. In step 6, where G3P is turned into 1,3-BPG, an NAD plus molecule is reduced to NADH. In step 7, 1,3-BPG donates a phosphate to ADP to form ATP. Then in the final step, PEP donates a phosphate to ADP to generate an ATP and a pyruvate. The final products from glycolysis are 2 net ATP, 2 NADH, and 2 molecules of pyruvate. Pyruvate will then move on into the next step, which is the pyruvate dehydrogenase complex, and then to the citric acid cycle. Here's back to the map. Highlighted in orange means that we're going to be in the mitochondrial matrix, and we're going to be starting with pyruvate dehydrogenase, the complex. The pyruvate dehydrogenase complex will link glycolysis to the citric acid cycle. The process converts pyruvate into acetyl-CoA, which will then flow into the citric acid cycle. The overall reaction involves decarboxylation and the transfer of an acyl group to CoA. These 2 main reactions will produce carbon dioxide and NADH. As mentioned, acyl-CoA will now be able to enter the citric acid cycle. The citric acid cycle is important for making the majority of electron carriers, such as NADH and FADH2. The citric acid cycle contains 8 catalyzed reactions, which mostly involve the oxidation of acyl-CoA, which will form CO2 and also will form the electron transfer molecules NADH, FADH2. For each acyl-CoA, around 3 NADH, 1 FADH2, 1 ATP, and 2 carbon dioxide will be produced. If we look at the picture I filled in, we can see some green boxes. These boxes include molecules that can enter the citric acid cycle from other pathways, which we will not cover in this video, but it's important to keep in mind that other things can come straight into the citric acid cycle. Moving on now, we are going to be looking at the electron transport chain, highlighted in red here on the map. This signifies that this is going to be happening inside the mitochondrial inner membrane. Let's get a zoomed in picture here. Now that we are in the electron transport chain, we're able to use the electron carrier molecules that we have been producing in other steps. NADH and FADH2 will be used to transfer electrons to generate hydrogen ions that will form a proton gradient in the next steps coming. For a quick overview, we're going to see how NADH will give electrons to protein complex 1, and FADH2 will give electrons to protein complex 2. These electrons will then be passed to various molecules such as ubiquitin and cytochrome C until they have eventually passed through protein complexes 1, 2, 3, and into complex 4. Complex 4 is then able to pass the electrons to oxygen molecules, which serve as the final electron acceptors of the electron transport chain. Oxygen is then able to react with protons that are generated to make water as a byproduct. Now that there is a proton gradient established, we can finally move on to the mechanism of ATP synthase, where the majority of ATP will be produced. Here in the dark red is the ATP synthase mechanism, meaning that's happening in or on the inner mitochondrial membrane. ATP synthase is able to use its large protein complex seen in the picture to create energy through chemiosmosis. It achieves this by harvesting energy from the proton motive force. Protons will follow their electrochemical gradient through the first protein complex, called complex F0, which causes complex F0 in the ATP synthase enzyme to start rotating. This rotation of F0 will cause the subunit F1 to rotate as well, which in turn causes a series of conformational changes within different subunits in ATP synthase. There are three main conformation changes. There are open, loose, and tight. Open allows ADP and phosphate to enter the active site of the enzyme. The loose allows ADP and the phosphate to slightly bind to the enzyme. And then finally, we get a tight squeeze where ADP and PI are forced together to form an ATP. Now we can look at the top left of this map, and we're going to be talking about how this all ties in to the nucleotide formation. So as we can see here, we have a PRPP, but how are these steps relevant to the production of nucleotides? One reason is that ATP is obviously necessary for the production of the nucleotides and eventually for DNA replication, but there is also another important molecule at play here, as we can see, PRPP. As PRPP is formed right here from G6P, which was one of the initial intermediates in glycolysis. PRPP is a precursor for purine synthesis, as well as being used for pyrimidine synthesis. PRPP is able to donate ribose phosphate groups, which enables de novo synthesis. So we have some ribose, we have PRPP, which we will see in a second, can go into produce some nucleotides. Here's the overview on the map so we can kind of see the flow of things. And we're going to take a look in this next diagram at the reactions of PRPP and how it is involved in the synthesis of nucleotides. There's a lot going on in this diagram, but we can see how we start with PRPP and end with IMP or UMP. They're not abbreviated on this, so I'll kind of, right here, we have UMP, and then down in the bottom right is IMP. These two different molecules will be very important in our final process, but we can also see in the drawings how we have a slow formation of rings as we follow the steps of the process. But after this step is important. This is just the formation of UMP and IMP. But after this step, PRP is able to be used just straight into being incorporated into nucleic acids and DNA synthesis. So these precursors here are able to immediately form ATP, CTP, UTP, or TTP, also GDP, or in regards to DNA replication, AMP, GMP, CMP, UMP, and TMP are able to be used as building blocks with DNA strands, which will then allow base pair binding with adenine, thymine, guanine, and cytosine. So as we can see, this PRPP is very influential when it comes to DNA replication, because without it, we would not have these basic precursors to building the DNA strands and allowing base pairing, which is important for our DNA, obviously. Now for our final picture, we're just going to be looking at what would happen if we're not going straight to DNA replication, and that is this diagram right here. We see UMP at the top and IMP at the bottom, and they're going through a series of reactions, but we're going to be mostly focusing on the ATP formation, because we want to see just a little alternate way of making ATP. So we can start with IMP, produced in the de novo synthesis that we saw on the last slide, and ATP can be produced by releasing a fumarate and then being phosphorylated until it is eventually an ATP molecule.